Hi, my name is Spencer Hale, and I'm a senior in biology here at Montana Tech. Today I'm going to be talking to you guys a little bit about HIV. We're going to be talking a little bit about what makes HIV special, the life cycle of the virus, and then we're going to be going into a little bit of a recent publication and how this recent publication and what they found can actually have an influence on the way that we possibly may end up treating HIV in the future. So HIV is human immunodeficiency virus, and what's interesting about it is it actually attacks the cells of our immune system directly, rather than attacking all the cells of our body. It is spread through contact with infected body fluids, and it's a reverse transcription virus, meaning that its genetic material is actually RNA, and it actually hijacks our cell's machinery to reverse transcribe its RNA into DNA, which then can be implemented into our genome. So up here, you'll see that I have CD4 positive and CD8 positive T cells basically divided. And these are two subsets of T cells that are generally referred to in immunology. Our CD4 T helper cells are basically those cells that will browse antigen-presenting cells, which are actually have MHC class II on their surface, and they're displaying antigen peptides on the antigen-presenting cells, which then the CD4 T helper cells will interact with. And if they actually match up with that peptide, will proliferate, differentiate. And CD4 T cells also will be important in the activation of CD8 T cells. The CD8 positive T cells are cytotoxic T cells, and those T cells are important for browsing MHC class I, which are found on all of our cells in our body. And usually MHC class I's are basically displaying either intracellular self-peptides or intracellular viral or pathogens like that. So HIV Life Cycle, we'll go into a little bit about that. So HIV actually infects our CD4 T helper cells. So it doesn't infect our CD8, but it will infect our CD4. It's going to basically bind to our T cell receptor on our CD4. It's going to fuse to that, and it's going to basically get itself into the T helper CD4 cell. It's then going to hijack our cell's machinery, and it's going to take our reverse transcriptase enzyme and convert its RNA into DNA. The third step is going to involve it's going to take that DNA and it's going to integrate it into our genome. So it's going to implement it into our DNA. Fourth step is going to involve transcription, and just like our body goes through normal transcription to make mRNAs, to make proteins, it's going to hijack our machinery again, which is and basically take our DNA as well as its viral DNA and make new mRNA, and then which can be further made into proteins. Step 5 is going to be the assembly of those proteins as well as some of the viral RNA that was just made, that will stay viral RNA, to make new virus. And then step 6 is actually going to be the virus pushing out of the cell. And that's going to actually result in death of some of our T helper cells. So we're going to steadily see a decline in some of our CD4 T helper cells as HIV progresses. And that's what's really important in this virus is normal individuals have between 500 CD4 helper cells and 1,200 CD4 helper cells per millimeter. But basically, once you get below 200 T helper cells per millimeter cubed is when actually you get the development of AIDS. And AIDS is acquired immunodeficiency syndrome. And what's important regarding AIDS is it's actually caused by HIV, and it's when our T helper cells just fall to a really, really low level. And usually, people don't actually die from HIV. They actually usually die from just common infections, because by losing our T helper cells, we lose part of our adaptive immune system. And by losing that, we can't respond to pathogens as well. So now let's look at this recent publication. The publication, it's actually a review article. It's the role of PKC-theta in CD4 positive T cells and HIV infection. So the nucleus and back again. And what basically this review article was looking at was it gave an overview of CD4 and CD8 T cells, where they're found in the body. But then it's specifically looking at what PKC-theta does in T cells and what we see in cells infected with HIV and the differences between normal T cells. So PKC-theta is actually a member of the protein kinase C family, and basically what happens when the T cell receptor binds to an MHC class II, since this is a CD4, displaying a peptide that matches, along with the co-stimulatory molecules of CD28 and CD3, you're going to get signaling, and the signaling ultimately leads in transcription factors in the nucleus, or basically being translocated into the nucleus of the cell, which will then result in different gene expression, including some cytokines like IL-2 being released and things like that. So they kind of went a little bit into the general PKC-theta signaling, but they also talked about other roles that PKC-theta actually has in a T cell. And I'll hit those really quickly, and then we'll go back to this. So PKC-theta is important in T cell energy and apoptosis, and the reason why it's important in apoptosis is if we have PKC-theta, it's actually important for phosphorylating our pro-apoptotic molecule BAD, which when that's phosphorylated, it's actually inactivated. So by having PKC-theta, we don't actually have apoptosis. By removing PKC-theta, we actually do get apoptosis. And also, PKC-theta actually aids in the development of Th2 and Th17 subsets, which those are all subsets of helper cells, T helper cells. But it's not as important in Th1 development, which is kind of interesting. And then the other thing they talked about was that PKC-theta is important in the development of Tregs, but it actually, after the thymocytes that have become Tregs, then actually PKC-theta is actually not good for the cell to actually be a Treg. So once it's a mature Treg, then it's actually going to decrease its PKC-theta. So let's look back over to here. So basically, the one pathway that PKC-theta is really important in the paper stressed was that it's going to lead through the IKK complex, and then it's ultimately going to result in Nf-kappaB transcription factor being added to the nucleus, which then, through gene expression, we're going to get IL-2 production. And IL-2 production is very important in T cell differentiation and proliferation. The other pathway that it's important for is actually when we get a signal through here, phospholipase C is going to cleave our inositol phospholipids into inositol phosphate as well as diacylglycerols, which is going to lead to PKC-theta. And what this study, which the review article specifically mentioned was a study by Sutcliffe et al., was that PKC-theta can actually itself be translocated into the nucleus of our cell, where in this cell is actually going to bind with a few other different proteins, including RNA polymerase II, MSK1, and LSP1, and it's going to lead to transcription. And what this is important for is they actually looked at PKC-theta in HIV cells, and what they found was that in HIV cells that we had a much higher production of PKC-theta relative to other cells. And what is kind of interesting is that this PKC-theta, it actually aids in the progression of HIV. And the reason it does that is because HIV has a Nef gene, and PKC-theta they found was actually a cofactor for that Nef gene, allowing it to actually be transcribed, which will actually allow it to be the virus to keep spreading and progression of HIV. So what they actually did was they decided, okay, well let's inhibit this PKC-theta. So they inhibited it and they actually saw a slight decrease and didn't completely stop, but a slight decrease in the transcription of HIV and the progression of HIV. So what they also did, which was kind of interesting, is they increased the amount of PKC-theta. And in vitro, what that actually did was it actually assisted in the eradication of virus-infected T helper cells. They did that basically by taking prostratin, which is a PKC activator, and it's a drug, and they can activate PKC-theta to cause an increase in it. What they don't know right now is whether or not this increase in PKC-theta would actually have the same effect in vivo, and whether or not it could have an increase in PKC-theta could actually have an impact on our regular non-infected cells, as well as Tregs and Th2 and Th17 subsets. So we still need to study more, and what the paper basically discussed is that more mouse models are going to be needed to see how it actually interacts in vivo. But this has a huge, huge impact on the way that we could actually end up treating HIV. We could be, whether we're actually decreasing the amount of PKC-theta, or whether we're increasing the amount of PKC-theta to actually cause the eradication of basically infected HIV cells. So thank you. I hope you enjoyed this video. I hope you learned something. Thanks.